So where we left off on Tuesday, whenever I did our quick 16-minute lecture on the cell membrane to kind of remind you about the cell membrane, I went over the details of it, but my whole goal was to end on why we call the model that we use to study the cell membrane the fluid mosaic model. And I introduced the term fluid, and then I introduced the term mosaic, and I wanted to bring those two together at the end of our previous lecture to put fluid mosaic together as the fluid mosaic model. Do you recall what aspect of this model is fluid? The phospholipid bilayer, yes. So this part right here is fluid. What part represents the mosaic? The proteins. Do you remember we had two classes of proteins? They were peripheral, which means that they were located where? On the outsides, right? We talked about the term peripheral. And then integral, which were integrated into that cell membrane. So we had discussed that. So we just looked at anatomy. Now we did briefly mention the function of a cell membrane, which is also called a plasma membrane, phospholipid bilayer, and biological membrane. What is the function of this? Yes, to regulate what goes in and out. So it's easy for us to look at this cell and say, okay, this is the cell membrane right here, and it regulates what goes in and out of the cell. But remember, by definition, eukaryotic cells contain membrane-bound organelles. So anywhere we have a phospholipid bilayer, so around the nucleus or around any of the organelles that are bound by membranes, it controls what goes in and out, okay? So it is easy just to say this is the only one, but I need you to know, as a college student, that this phospholipid bilayer is present around a lot of these organelles as well. If I were talking about a prokaryotic cell, there would only be one plasma membrane, and it would be around the outside, because prokaryotic cells lack membrane-bound organelles. What's a prokaryote again? A bacteria. The only thing on earth that's prokaryotic is a bacteria. Yes. Fantastic. So my focus today is the function of the cell membrane. And if the function is to control what goes in and out of the cell, we need to talk about how things go in and out of the cell. That's our focus. Okay? So there are two ways things can go in and out of the cells. Big picture. There is passive and active transport in and out of the cell. Before I take this into anything context-wise that has to do with biology, I just want to pull the common sense card. When you think of the term passive compared to the term active, how would you describe something that's passive? Slow. If you have a passive personality, what do you tend to do if things are going on around you? You're just like, Now, if we referenced active, there would be energy. You're doing something. You're moving. Outside of the context of cell membranes, passive and active, passive basically says you don't use a lot of energy, if any, and active says you do. So I'm going to use what we already know to kind of help us through these terms. So passive transport, I'll say, uses no, when we say energy, what am I talking about? ATP, fantastic. No ATP, and we'll say active, we use ATP. Okay? Very well. And as I'm looking, this is, of course, my first slide on passive transport, and it's clearly labeled at the top. It defines the term diffusion. So diffusion is a type of passive transport. Diffusion. So I'll start with that. And next to diffusion, it says It defines it. It says this is the net movement of molecules from an area of high concentration to an area of low concentration. It's the net movement of molecules from an area of high concentration to an area of low concentration. It mentions the concentration gradient, and I'm going to come back to that. But I want to go through the definition. I want you to understand what that means. If this were my cell membrane, and I was talking about concentration, I want you to know what concentration means. Concentration refers to the number of molecules. Concentration refers to the number of molecules. So right here, I would say that on the left side of the cell membrane, there is a high concentration of molecules, and on the right side, there is a low concentration of molecules. So high concentration, low concentration. I want to put something concrete with those terms so you can picture them. So concentration is the number of molecules. Now, for diffusion, it says that these molecules move from an area of high concentration to low concentration. An area of high concentration to low concentration. So in this example here, are these molecules going to move from the left to the right or from the right to the left?
Right to left.
They're going to go from an area of high concentration to an area of low concentration. So they're going to go from the left side to the right side. They're going to go from here to here. High concentration to low concentration. When I teach this to my 9th grade freshmen, I use a waterfall because this is a concept that you don't see every single day because a cell's microscopic. You're not just seeing this happen. I use a waterfall. If you are at the top of a waterfall, okay, and that waterfall is being fed by this huge source, whatever it is, because the waterfall just continues to flow. If you're at the top of that waterfall, if you jump into that waterfall, where there's a high concentration of water, where are you going to go naturally? Straight down. And you're going to go to the pool wherever that waterfall ends, which has a lower concentration of water in comparison to what was at the top, because the water continues to flow down. How much work did it take you, bless you, to get from the top to the bottom? None work. No work. Like it just happened because there was a current going and you jumped in and you just followed the current. You went from an area of high concentration to an area of low concentration. And it didn't require any energy from you because it just happened naturally. Does that make sense? Okay? So, it mentions the high concentration, the low concentration, the no energy, and now I want to touch on the term concentration gradient. It says that with passive transport, we go with the concentration gradient. We go with the concentration gradient. This means that we're going the natural direction, an area of high to an area of low. So going with the concentration gradient is going to from an area of high concentration to an area of low concentration. Okay? Now, this slide right here talks about our first type of diffusion. Our first type of diffusion is simple diffusion. Simple diffusion. There's a couple of things I want to point out on this diagram, and then I'm going to add to what I have over there. First of all, as I've mentioned countless times before, when you have a diagram at the collegiate level, it's supposed to be conveying information to you. It wants you to interpret it. Understand what it is I'm showing you. So if I gave you this diagram, I'm going to pretend like I'm you, and I'm trying to figure out what it is that this diagram is conveying to me. Okay? I can see that there are labels, so I'm trying to figure out where I'm at. It says here I have a lipid bilayer, in parentheses, plasma membrane. Nice, because anyway. So I have my lipid bilayer. I can see that. Here it says this is extracellular fluid, and this is cytoplasm. Which one is inside the cell? The cytoplasm. The cytoplasm is inside the cell. The extracellular fluid is on the outside of the cell. So that's the outside of the cell. This is the inside of the cell. Okay, so that tells me where this cell membrane is. It's the one all the way around. And then here I'm looking at the molecules. It appears that right now I have a high concentration, and down here I have a low. And I continue to have high, but I can see that some of them are just going straight through and to a lower concentration. And now I have about an equal concentration, and it's showing me, if I pay attention, that this is taking place over time. So it's showing me that it's changing. Where I originally had a huge high concentration here, low here, over time the molecules went from an area of high to low until they were about equal. Okay? So that's what that diagram is telling me, or showing me, or that's what I would get from that. This is demonstrating simple diffusion. Simple diffusion shows these molecules just going straight through the plasma membrane. Just going straight through. I taught you something in Chapter 3 about plasma membranes that we've discussed over and over again. They have two characteristics. They're hydrophilic and phobic. Looking at this distribution, is there a larger portion of this cell membrane that's hydrophilic or hydrophobic? Hydrophobic. This part right here is much thicker, the lipids, right? The content I was teaching you then was steroids. And I said, when you take a steroid, because it's lipid-based, what can it do when it gets to your cell? Versus, 100%, versus a protein or a water-based substance. A steroid is fat-based, it can go straight through. It can go straight through anywhere. Because it's a lipid and lipids like lipids. In simple diffusion, the molecules that can go through are soluble in lipids. They're what we call lipid soluble. So they get along with lipids. As a result, they can enter the cell wherever they want because they're lipid soluble, so they just come right in. Could water do that? No. When water tries to come in the cell says, hey, I'm fat. I don't want water. Not like it's fat. You know what I'm saying. It's hydrophobic. I'm afraid of water. I don't want water. Okay, so adding to our chart over here, I want to put that under diffusion, the first type is simple. And the type of molecules that can just go straight through are what we refer to as lipid soluble. These, the ones that can go straight through, are lipid soluble. I'm pretty sure I spelled that wrong. Oh, I didn't. Good. Simple diffusion, lipid soluble. Okay? Now, next type of diffusion, facilitated diffusion. And before we go into exactly what facilitated is, let's talk about the term facilitate. What does that mean? To facilitate anything, like teachers are considered facilitators of knowledge. So you're supposed to be conveying knowledge, but it doesn't matter how much knowledge I have unless you're open to wanting to learn that, to receive that information. So when you facilitate something, you kind of guide it. Or here's the correct pathway, okay? Or you oversee it. In facilitated diffusion, My pictures are slightly different, so let's look at what we have in front of us. I clearly see I have a phospholipid bilayer, but now I have this crazy structure right here. What is that crazy structure? It's a protein. Is it an integral protein or a peripheral protein? Integral, and I know that because it goes all the way through. Fantastic. It's embedded in that plasma membrane. Now, I can see there's molecules here, and this diagram is clearly labeled cytosol and outside the cell. So I know that this is inside and this is outside. When I look at these molecules, I don't need to count them, but I can generally see that there's a higher concentration here and a lower concentration here. And in which direction are they going? How do you know? The arrow clearly indicates it's going from an area of high concentration to an area of low concentration. But look at the molecule that it is. This is water. And just a moment ago, we said if water shows up, the cell's like, I'm afraid of water. So the only way water can get in is if it has to go through a protein. It has to find a door that says, hey, come on in, you can come in this way. So facilitated diffusion still goes from high to low, but it doesn't just go anywhere. It has to go specifically through a protein. It has to go specifically through a protein. So it's still going from high to low, but because it's not lipid soluble, it has to go through a protein. Before I add to the board, I want to point out something on the second diagram. People naturally think when you say high to low, you're always going to be going from the top to the bottom. I'm just saying we're going from a high concentration to a low concentration. So when I analyze the second diagram, I have my phospholipid bilayer and I have my integral protein. Where's my high concentration? At the bottom in the cell. And where's my low concentration? Outside the cell. And which direction is it going? And we know that because there's an arrow. So it's still going from an area of high concentration to an area of low concentration. Does this require any energy? No. It just happens naturally. So I don't want you to always assume when you're looking at a diagram that the high concentration will always be at the top, because it's not. Okay? Just know what it is. Look at the picture. What information is this picture giving me? Okay? So on facilitated diffusion, which is my second type of diffusion, this only takes place, so facilitated. Non-lipid soluble molecules. Okay, now, in a big picture type situation, it may be that something is lipid soluble. There could be some exceptions to this, but you and I aren't covering that, okay? So those of you who go further, if you come back and look at your notes, because if you're going to study science, I would say keep all your notes. There are some exceptions, but we're not going to cover those in here, okay? So simple diffusion is lipid soluble. Facilitated diffusion would be something that would be non-lipid soluble, like waters or proteins or vitamins, things that aren't fat-based or aren't lipid-based. Okay? And facilitated diffusion requires the use of a protein because those molecules cannot just go straight through the cell membrane. In this diagram here, this is an example of facilitated diffusion. I have my phospholipid bilayer, and I have my integral protein. Where's my high concentration? On the top. It is on the top. And the low concentration is on the bottom. The difference here is I don't have a whole bunch of molecules going all together at the same time. I have just one at a time. One at a time. And that molecule will enter here, where it's verified, and then that protein will change shape, fold, and allow for that molecule then to enter the cell. What is this protein going to do next? It's going to repeat it. It's going to keep going over and over again. So this might be for a molecule that's much larger and doesn't easily flow through small protein channels, okay? When I teach this to my freshman 9th graders, I say it's like when you have an invitation-only party. When you have an invitation-only party and you require that everyone that comes in has an invitation, you have to verify each person has an invitation when they come to the door, okay? Because when you say it's invitation only, I don't care if you're popular or not, everybody wants to come to the party now because only a select few were invited. So if you were unpopular, you just became popular because everybody's like, oh, I'm going to that party. Are you going to that party? And the people who didn't get an invitation are like, I'm not going to that party, but they still show up trying to get into that party. But it's invitation only. So as you come through the door, we're verifying that you are indeed the recipient of that invitation. And then if you are, go ahead and come in. And then the bouncer checks the next person and so on. And if you have a bouncer at your party, that's crazy. I mean, you must be really awesome. I don't even know. But carrier-mediated diffusion is a type of facilitated diffusion, okay? Osmosis, another type of diffusion. Osmosis is another type of diffusion. But does anyone know what osmosis strictly deals with? Water. If it has to do with osmosis, if osmosis is being used in your question or sentence, it's dealing with water. If it's salt, it's diffusion. We just say diffusion. If it's sand, we just say diffusion. If it's water, we say it's osmosis. If it's Oreos, diffusion, right? Because it's not water. Whatever it is, if it's not water, it's diffusion. If it's water, it's osmosis. Okay? I'm going to explain this diagram in just a moment because this pertains to the lab that we're going to do. But I want to discuss these terms with you. A selectively permeable membrane. Selectively permeable membrane. What do you think that means? Only selective things can get through. Permeable talks about what can get in and out. So if it's selectively permeable, only certain things can get in and out. What if it was non-permeable? Nothing can get through. What if it was freely permeable? Everything can get through. Do you see how that term applies? Permeable. Okay? I also put, so selectively permeable membrane, it may be referred to as semi-permeable. So I wanted you to know that those two are the same thing. Those two are the same thing. All right? Now, again, we're discussing osmosis, the movement of water from an area of high concentration to an area of low concentration. So let me describe our setup here, and this is going to be us next week. So we have our beaker of distilled water, and then we have a thistle. You and I are not going to use thistles because they were breaking. We're going to use dialysis tubing, but we still have the same setup. Inside the thistle, we have a glucose solution. What do you think a glucose solution is? Sugar water. Yeah, it's just sugar water. But we're trying to be fancy because we're in science. So we're going to mix in a glucose solution. Okay? Let's just be silly like that. Anyway, so glucose solution is just sugar water. So we have distilled water and we have sugar water. And then we have this semi-permeable membrane, which is fancy for a hole with bags in it. A bag with holes in it. I said that totally backwards. A bag with holes in it, like little, you make some small little holes. But we call it a semi-permeable membrane because it is technically semi-permeable. It's a bag with holes. And we're going to wrap it around. You're going to have your dialysis tubing, which is also semi-permeable. Now, so here's our general setup. Taking this right here and blowing it up, kind of putting focus on it. So we have our glucose solution, our sugar solution. Okay, we have our semi-permeable membrane or selectively permeable membrane, same exact thing. And you can see with my distilled water here, these arrows are indicating movement. In what direction is the water moving? Up. And it's a bunch. How do I know it's a bunch of water moving up? the arrows are thick. Okay, and it shows some small movement here, and I'm going to show you what that is in just a second. It's still water, but it's moving in the opposite direction. So there's a whole bunch of water that's moving from a high to a low concentration. So now I'm going to take this part right here and emphasize it. So here's inside my glucose solution, and I know that because I have water, but I also have glucose. So I know that this is inside. Here's my selectively permeable membrane, little holes in the back, and here's my water. This large arrow tells you the water is, for the most part, moving up. And these smaller arrows are showing you that there is some movement, and some of that water is going down. But the majority, the net movement, the most water is moving up. Now, if you were just comparing the number of water molecules here to the number of water molecules here, which one has the higher concentration? At the bottom, there's a higher concentration here, a lower concentration here. So water, process of osmosis, goes from an area of high concentration to an area of low concentration. Why is that sugar not moving? Why is it not coming down? Because that membrane is selectively permeable. And guess what can get through? Water. Not anything else. So sugar's not getting through. Salt's not getting through. Sand's not getting through. Nothing else is getting through. This is selectively permeable just to water. That was the only thing, the selective thing that could get through. So on my diffusion, I also want to add osmosis. And osmosis specifically refers to the diffusion of water. Fantastic. Okay? Questions so far? It is, yes, because it's using a protein to go from high to low. It's a type of facilitated diffusion. Yes. Fantastic. What other questions? Okay. Now, still on osmosis, because we're talking about the movement of water, we're going to discuss the topic of tonicity. Okay? And this is normally something that in a biology class you go over really quick, like you're like, okay, tonicity, hypotonic, hypertonic, and you move on. But I'm going to tell you that it wasn't until I was way into college that I actually had an instructor explain it to me, and I don't want to be that instructor that kind of skips over this and expects you to know it. So I'm going to explain it because I know how the questions are going to be asked, and I want to make sure that you know how to interpret them. Okay? So tonicity is the topic, and I'm going to sketch out some beakers for you. So beaker 1, beaker 2, and beaker 3. And within my beakers, I will have cells. Okay, I'm going to do hypotonic first. A hypotonic solution. Hypotonic solution. In this case, I have a high concentration of water outside my cell and a low concentration of water inside my cell. And outside my cell, I have a low concentration of solute, and inside my cell, I have a high concentration of solute. Before I move further, I want to make sure you understand that concentration means the number of molecules, okay? And the term I really struggled with when my instructors were going over this was solute. Like, I don't know what you mean by solute. Solute can be salt, it can be sugar, it's anything that's not the water. Okay, so it could be sand, it could be Legos, it could be noodles, but it's whatever it is that you're trying to dissolve or mix into that water. Okay, so for this example, we'll just say that the solute is salt. That way, if you write that down in your notes and you're going back over it, you have an idea as to what's going on. So, in a hypotonic solution, the actual solution itself that I've put my cell in has a high concentration of water and a low concentration of solute. The solution does. The cell has a low concentration of water and a high concentration of solute. Do you see what I'm saying right now? So far, you can interpret what we have. Because this is what the questions are asked. They're going to ask you about the water, and then they're going to ask you about the solute. So I want you to know what you're dealing with. Now, with respect to water, which direction will the water move? Into the cell or out of the cell? Why will it move into the cell? It's going from where there's a higher concentration of water to a lower concentration of water. So you are exactly right. That water is going to move right into that cell.
So basically it's full of water.
Yes. Can you tell me what this cell is going to do? It's going to swell up. It's going to get bigger. So the cell will swell. And a quick memory tool, I didn't come up with this. It was a student a couple semesters ago. She said, I remember hypotonic because hypo and hippo. So it swells up big like a hippopotamus, like so. Hypo, hippo. And that makes sense to me. So I pass on those ideas if I hear them, and I think that they would help other people. So the water moves into the cell. You are 100% correct. It goes from an area of high concentration to low concentration. When we talk about it with regard to the solute, the water moves from an area of low solute concentration to an area of high solute concentration. I am truly saying the exact same thing, just from a different perspective. Like, you know how they say there's two sides to every story? From the side of the water, water's moving from high to low. But if I'm looking at it from the perspective of the solute, the water's moving from where there's a low solute concentration to where there's a high solute concentration. I'm not changing the direction of the water. It's still going in. I'm just telling you a different perspective. You need to understand both. I could understand the water part, but when you put the solute part in front of me, I was like, what? Like, are you trying to mess with my head right now? It's truly, they say the same thing. It's just who's telling the story. So if it's the water, I'm moving from high to low. And if it's the solute, the water is going from the low solute concentration to the high solute concentration. Yes? Everybody okay with that? Okay? Next situation, hypertonic. In a hypertonic solution, we have more solutes outside the cell in the solution than inside the cell. And the concentration of water is higher in the cell than outside the cell. So in this case, I have a whole bunch of salt and just a few water. And inside the cell, I have a lot of water and a little salt. This is you, and this is the ocean. This is exactly what happens to you when you get into the ocean. which direction is this water going to move? It's going to move out of the cell. It's going to go from an area of high water concentration to an area of low concentration. What's going to happen to that cell? It's going to shrink. It's going to shrivel up. That's what happens to you. People are like, what? The ocean has far more water than I do. The ocean does have a lot of water, but it has a higher salt concentration than you do. So when we put you in that salt water, the salt draws the water out of you. So even though you're in the water all day, you get out of the ocean and you're dehydrated. How do you feel? Tired. Not the same way you would feel if you were swimming in your pool at your house. If you have a pool at your house, I don't know, I didn't want to assume, sorry I shouldn't have said that, but a pool somewhere, okay, where it's not salt water, unless you have a saltwater pool. Anyways, whatever, you get what I'm saying. From the perspective of the water moves out. It says I'm moving from an area of high water concentration to low concentration for water. If I'm talking about the perspective of the solute would say, once again, the water is moving from an area of low solute concentration to an area of high solute concentration. Said the exact same thing, just from different perspectives. Okay? I don't have a memory tool for Hypertonic. That was somebody else's. I don't want to take credit for it. Okay, last one, isotonic. Isotonic says we have the same amount of water outside the cell as inside the cell, and the same amount of solutes outside the cell as inside the cell. Just so you know, water still moves, but what direction does it move? It's going both ways. Like, yeah, it's just staying balanced. It's staying balanced. And I use the term balanced because I don't want to use what word? I don't want to use the word homeostasis. What word applies here? Isotonic. What type of solution is it when it's balanced? Equilibrium. Why is it not homeostatic? It's not living. Homeostasis applies to something that's living. Okay? So, hyper, isotonic. Understand the distribution of the water and the solute and be able to explain which direction the water moves. Okay? From both perspectives. Yes, ma'am.
Can't you remember that I just said the word roots?
Yeah, yes. And so it makes perfect sense. It makes perfect sense until you read the question and it's like the concentration of solutes is greater or low. I mean, you're like, oh. So you are 100% correct. The word roots do help you learn what the word does mean, and you can kind of distinguish that or decipher it. But when it starts to ask about the movement of molecules, I want you to be solid on that. Okay? I wanted to take a little bit of extra time to explain that so that you knew. But yes, ma'am, you are 100% correct. The word root does reinforce this content. So let's look at this situation, hyper, hypo, and isotonic in a plant cell. Too far. Stop. In a plant cell. First of all, this is a super happy plant cell. The term turgor pressure, this is the second time I've exposed you to this term. Turgor pressure is when that vacuole is full and it has so much water it pushes up against the cell wall. What does this plant look like in real life? Here's its cell, but what does it look like? Is it happy? Is it sad? This is a happy plant. It's got a lot of water. It's standing up nice and pretty. All the leaves look great. It's a happy plant. Plants love hypotonic solutions. Plants love hypotonic solutions. Okay, when you buy plants that have already been cut, like roses or whatever at the grocery store, they have a solute packet, which has their magic mix, whatever it may be for that vendor. But you add it to the water, and it helps the plants. It helps create a hypotonic situation so the plants stay prettier longer, as opposed to just putting them in your fresh water. This plant is healthy, but it is not exactly happy. That vacuole is not as full as it could be. Okay, so this is isotonic. Hypotonic, isotonic. Plants are still alive here and healthy here. They're just not as happy as they are in a hypotonic solution. Now this plant here in the middle, I took this diagram from a plant that was at my house, because look at, what's the problem here? The vacuole has very little water. Probably because I didn't water it. And I probably didn't water it because it didn't tell me it needed to be watered. It's its own fault. Okay? I already told you, my kids don't eat unless they say, are we going to have dinner? I just study all the time. I'm a huge nerd. Poor kids. It's like 11 o'clock at night. Mom, what are we doing for dinner? What? Y'all haven't eaten? No? Where's Dad? At the fire station? Well, cereal? So, the thing is with these plants here, this plant cell, yeah, it hasn't been watered, but what is this structure here? This is cell wall. And the cell wall is like the external skeleton, right? So when a turtle dies, does its shell shrink? No. So when this cell is dying, the cell wall stays there, but inside starts to deteriorate. What does this plant look like? It's brown. It's probably like wilted. I could probably take a picture of it at the house and bring it and show it to you. But it's just not happy. What is this? It's pointing, it's going in. What is that structure that's pulling in? It's the cell membrane. The cell membrane is pulling itself off of that cell wall because this cell is in distress. It's like, oh my God, I'm dying. Well, you wouldn't be if you would have said, oh my gosh, water me. And I would have watered you. But this cell membrane is pulling itself off. The term we use to describe this is plasmolysis. Technically, it's plasmolysis, okay? But I'm like so your plasma membrane and lysis. So it's being broken down, okay? Plasmolysis is this situation. The cell wall has stayed there because it's robust, and the plasma membrane is kind of shrinking in and pulling itself apart. This cell in a few days is going to expire if someone doesn't water it. So plants prefer hypotonic. They'll be okay in isotonic, but hypertonic is not ideal for plants. Now, that's plants, because animals, it's a different situation. These are the red blood cells found in animals like you and I. Okay, in a hypotonic solution, our red blood cells would swell up really big. Let me preface this. In case you didn't know, this is what your red blood cells should look like. They should be like, I always describe in anatomy, they should look like a donut without the hole punched out. Okay, and their shape is amazing, and it intricately works to function extremely efficiently. This is supposed to carry oxygen throughout all the tissues in your body. This is a red blood cell. This is what it should look like. When you are overhydrated or there is too much water, your cells will swell up into the point where they lyse. In animals, we call that hemolysis. Hemolysis. And the cell will swell up until it lyses or ruptures or bursts. Hemolysis. And that's specifically, hemi is blood. Anyways, here is you dehydrated. Again, the shape has changed. It's shriveling up. You can see the cytoskeleton poking out. Like this is an unhealthy cell. Is it carrying out its function? No. And in fact, when you are dehydrated like that, how do you feel? Very tired, very weak, and the longer you go with being dehydrated or not getting enough hydration, you start to forget things, you become disoriented until finally you shut down and expire. Okay? I've had someone in the first class say, so how would you become overhydrated? This is not something that you would naturally expose yourself to. You would have to drink like 2 to 3 gallons of water within a 1 to 2 hour period to do this to yourself. That's a lot of water. You do need water, yes. But that much water, it hurts your kidneys, it hurts your organs, because there's so much water, and it takes the ions in your body, and it kind of decreases their saturation level, and so the concentration of them decreases with respect to the water, so you're not conducting electricity. Slowly your organs start to fail. There was a case at colleges where they were rushing for fraternities, and that's what came up in the news a couple of years ago. Whatever they were making their pledges do, they had to drink an excessive amount of water, and it actually killed one of them, 2 of them, I believe. So, because you just can't handle that much water, okay? When we do our lab next week, we're either going to have horses or goat blood, depending on what vet tech gives us. But we're not using human blood, because nobody wants to share diseases, okay? Nobody says, hey, I have a disease, before they let you touch their blood. Nobody does that, okay? But we're going to use an animal's blood, other than ours, and we're going to add a bunch of water, and you'll be able to look at it under the microscope and see the cells get big. And then we're going to take salt water, and we're going to put salt water on it, and you're going to see that same cell go and shrivel up. What was your question?
With the hypotonic cell,
Hypo.
Is that what happens when you drown?
No. When you drown, that's a great question. When you drown, you've gotten water into your lungs. So where you're supposed to respire, and it's just supposed to be air, carbon dioxide, and oxygen exchange, you have water. And because there's water there, you can't exchange gas, and that's what drowning is. Your cells don't become hypotonic when you drown.
If you stay, like, for example, let's say that you are lost in the ocean, or whatever, or in a lake. You drown, and then you stay in the water, like at the bottom of the lake.
So you will swell, yes.
Okay.
Yes. That's not necessarily just your lungs. That's all your body tissues taking on water. Good question, though. Good question. Okay, questions? Any additional questions on tonicity?
What did you say again? The hemolysis?
Hemolysis is in animals it's a hypotonic situation where that cell swells up and it ruptures. That hemi is talking about blood. So this happens to be red blood cells, so we would call it hemolysis. Additional questions? Okay. So if it's not passive transport, it has to be active. And active doesn't have near as much to it as passive does. If it's active, we already know it's going to require ATP. And instead of going from a high concentration to a low concentration, in this situation, they're going from a low concentration to a high concentration. So going back to my waterfall example, I was at the bottom, and I thought to myself when I got down there, man, that was really fun. I want to do it again. Am I just naturally going to make my way back up there? Yeah, no. It's going to take a little bit of work. Like I'm going to have to get out and climb. That's going to require some ATP. So an active transport, we already know it uses ATP, but it doesn't go with the concentration gradient from high to low. It actually goes against the concentration gradient. And that means it's going from low to high. Means it's going from low to high. So on this diagram that I'm about to show you, I'm going to kind of walk you through and help navigate what's going on. First of all, can you see the mouse? You can't see the mouse. Let me do this. 1 second, please. Now you can see mouse. Perfect. Okay, so the things that I would point out here, if I was looking at this diagram for the first time, what is this structure? That's a phospholipid bilayer. What is this structure right here? It's an integral protein. So I'm navigating myself. Where is this phospholipid bilayer? This is outside of it. This is the cytosol. So this phospholipid bilayer must be the one on the outside. Okay? Because it's saying outside the cell and cytosol, which cytosol, in case you forgot, is that jello type structure on the inside. Okay, for real quick clarification purposes, I had a student say, is cytosol the same thing as cytoplasm? And I covered that in a previous lecture, but once again, cytoplasm is anything that is outside the nucleus, but inside. So all of this is considered cytoplasm. Cytoplasm has two components, the actual organelles, the structures, and the cytosol, which is the jello type structure. It keeps everything separated. So cytosol is part of the cytoplasm. Cytosol and cytoplasm are not the same thing. Okay? But, so I'm on the outside plasma membrane. Here's outside, here's cytosol. Now, I'm going to start with this one. First of all, Na+, this is a sodium ion. It's a cation. Okay, and we've studied cations already. So this is sodium, and it's telling me in this situation I have a high sodium concentration outside the cell, and inside the cell I have a low sodium. Do I have a bunch of molecules drawn on this diagram? No, but it's telling me, for reference point, this is high and this is low. Over here, K is potassium, so I have potassium ions. It's telling me that outside the cell, my concentration of potassium is low, and inside the cell, my concentration is high. Now, what is this right here? What are these? Molecules of what? How do you know they're Na or sodium molecules? It clearly says that. It says, bless you, it says you have 3 sodium ions. Which direction are they going? And I know this because there's the arrows. It's telling me I'm going up. So am I going from a high concentration of sodium to a low, or from a low to a high? I am going from low to high. Is that passive or active? Do you see how I haven't even gotten to the other side, but I can just tell, the other diagrams, I can just tell by the information given to me right now that this is going to have to be active transport because it's going from a low concentration to a high. Oh, look what's right there. It's ATP. I'm going to tell you, and I'm going to say this, and you will probably, some of you are going to listen. And some of you are not, but I specifically, throwback real quick, I specifically told you when we did fructose and glucose and you built the ring structures and the linear structures, these are going to be on the lab practical. If it's a pentagon, it's fructose. If it's a hexagon, it's glucose. I didn't know which one he was going to put on the lab exam. He put fructose. Do you know how many of you wrote glucose? I was like, I know I said pentagon, but not a big deal. Not a big deal. So I'm going to tell you one other thing that hopefully will help you answer a question right. And if you're really paying attention, you'll get it right. And if not, well, you already know. All right? So whenever you see this little burst right here, guess what that means? Yeah, it's active. It needs ATP. It needs energy. And my drawing right here is pretty much identical to this guy right here. I mean, not identical, but it's close. If you see that, active better be your answer. If you see that. Okay? And I'm telling you that because I know it's going to happen. I'm telling you, you get your little burst of ATP there, it's active. Okay? But you already knew it was active just based on the fact it was going from a low to a high concentration. So here you can see, just as you hypothesized, it was going from low to high. It used ATP. That was specifically referencing the sodium, and now we're going to deal with the potassium. This is taught separately for this demonstration, but in real life they happen at the same time. Okay, so I'm just telling you that, so that same molecule of ATP would have been used for both. But here I have 2 potassium ions, and outside the cell, was there a high or a low concentration of potassium? Outside the cell, there was a low concentration of potassium. Inside the cell, there was high. What direction is this potassium going? It's going into the cell. So is it going from high to low or low to high? It's going from low to high. It's still active. It's still active from low to high. And you can see that that's occurring there. Again, it would have used that same ATP from the other because they're happening at the same time. Okay, just so you know. And then that potassium comes in. Okay? I wanted to go through that slowly instead of throwing it all up there at one time. I wanted you to see how you can distinguish between active and passive transport just by using the information you've been given. Now, here's one where you don't see the burst of ATP. But if you know the direction, you know the answer. I'm going to pretend like I'm you and I see this diagram on my test. Okay, I can see that. I'm going to start here because I read left to right, so I'm just going to start here. It says driving ion in high concentration. I truly don't know what it means by a driving ion, but I do know what high concentration means. Okay? And I can see that it's pointing to the red, so I'm going to follow the red, and I see that the red is going down into the cell. And down here it says driving ion. Again, I don't know what that means, but in low concentration. So I know it's going from high to low. Thank you. You already know if it's going from high to low, it's passive. Okay? All right, cool. Driving ion, still not really sure, but I see that the red is going from high to low. I'm going to move over here. Transported solute in low concentration. So transported and driving, so now I'm kind of thinking like this one might be the lead and this one might be the passenger or something like that along those lines. But I see that it's going from a low concentration, and I know that it's going down because of the arrow, to transported solute in high concentration. So it's going from low to high, which automatically says it's active. Let me tell you what this is. When you got to the bottom of that waterfall, your little brother went with you, okay? And you were both like, gosh, that was so much fun. That was so much fun. And you said, I'm going back up there. And your little brother was like, well, I want to go too, but I'm tired. So what are you about to have to do for your little brother? Yep, piggyback. He's like, can I ride your piggyback? And he's not doing any work, but you're going in that direction. So is he expending any energy? No. Okay, he's just going along with that. All right, so it's still considered active transport if it's going from low to high. It's going from high to low, it's passive, low to high. Again, there's no ATP burst, so that wouldn't be the first indicator. Okay? Here, I have the same thing going, but instead of going in the exact same direction, they're going in opposite. And I have my driving ion concentration going from high to low, and my transported ion is going from low to high. Okay? So it's just knowing the directions. You know the directions. You can tell me what type of transport it is. Exo and endocytosis, our last topic. You already know what these are. Exo tells you it's going in which direction? Out. And cyto is cell. So exocytosis means it's going out of the cell. Out of the cell. Okay? And I told you that when a cell was transporting stuff within it, it would use these small transport vessels called vesicles. Fantastic. real quick. And you're going to need to know this for your test. What structure within the cell creates proteins that will be sent out for export? The ribosome does make proteins. Fantastic. What are they, free or fixed? Fixed. The ones that are going to be sent out are fixed. What are they fixed to? And I told you when I explained this, I was like, we can't feel it, but when we look at it through the microscope, it appears to be like bumpy, rough, endoplasmic reticulum. Yes. Okay. Yay. So the ribosomes make the protein, right? But it's the fixed ribosomes, and they're fixed to the rough endoplasmic reticulum. Those proteins will be sent out of the cell. Okay? Do you remember the place that they have to go before they get out of the cell? Yes, the Golgi apparatus, the post office. Because they're going to be shipped out. You got to know this for your test. They go to the Golgi apparatus to be modified, sorted, and packaged for export. So what we have happening here is we have our vesicle. Our mail truck is taking these proteins to their destination. And when that vesicle hits the cell membrane, does the vesicle itself leave the cell? No. No, I mentioned that the vesicle will then fuse with that cell membrane because the vesicle is just a phospholipid bilayer. It'll fuse with that cell membrane and it'll dump its contents out, which you can clearly see is happening in this diagram. So it's full of proteins, it gets to the plasma membrane, and it fuses with it and dumps its contents, and that vesicle does not leave itself. Exocytosis. The opposite of exo would be endocytosis. And endo tells you it's coming in which direction? Into the cell. Fantastic. Okay? And we discussed for endocytosis, and these diagrams can be a little bit confusing if you don't pay attention to the arrows. So I have my inside of my cell, outside I have this solution, and here's my plasma membrane. That solution is trying to come in, and it can't just pass right through, so the cell membrane creates a vesicle. And that vesicle will then transport that substance or solution into the cell. So it was here on the outside, it folded in the plasma membrane, and now it's carrying it to its destination. Endocytosis. What we have here is the exact same process, but you have to have a key to get in. So we call it receptor mediated. Let me give you an example of this. In order to get into your cells, viruses have to have a key. So there's 100% a chance that we could put virus in this room, and that virus would have the key to 3/4 of your cells, or 3/4 of our cells, so like 75% of us would be infected with the virus, and there would be a percentage of us that wouldn't because the virus didn't have the right key for their cells. The flu virus, if Araceli comes next week and she's got the flu, and she's coughing and sneezing and doing all that stuff, and we're like, oh my gosh, I cannot believe she came to class. But that next weekend, I start feeling sick. It's because the flu virus she had the right keys to get into my cell. But she was sitting right next to Andrea this whole time, hacking up. And Andrea comes next week, and she was never sick. It's because that virus didn't have the right keys to get into her cells. So we all have unique keys based on your genes, your DNA. Your DNA decides what keys you have and what you're going to be infected by. So this is still endocytosis, but you have to have a key to get in. Okay, so that's the only difference here. On endocytosis, we can break endocytosis into two forms, pino and phagocytosis. Pino is cell drinking. And so I remember the wine, Pinot Grigio. I'm like, oh, the cell is drinking. Okay, in this case, in pinocytosis, the cell only brings in fluid. So when it's bringing something in, it only brings in fluid. Phago is whenever the cell brings in something larger, like a big bacteria or a complex molecule of some sort. because it's larger and it's not just liquid, we call it cell eating. Okay? So pino and phagocytosis are both endocytosis. They are. It's just subdividing. Is it bringing in fluid? So we call it pino. Is it bringing in something else, something large like a bacteria or a glucose molecule or something like that? That's phagocytosis. And the last thing I want to explain, it's a type of phagocytosis. This handsome-looking organism right here is what we call an amoeba. An amoeba is unicellular. What does that mean? It's one cell. So can you see this with your eyes? No. But when amoebas, look at that body. I mean, it clearly works out to get that shape. But when they want to eat, they don't have arms and legs. They throw themselves at their food. It's quite disgusting in all honesty. Okay, and if there are YouTube videos that show it, so I suggest, you watch it, but I know you won't. But you should. Because the first time I watched it, I was like, no way. They kind of just like, and you're like, oh. And there's sound effects in some of them, so it helps bring it to life. But when I saw it the first couple times, it made me think of those slappy hands you get at the, the vending machines, those sticky slappy hands. Okay, I hate them, but Isaiah loves them. And so he's like, do you have a quarter? And the first time they stick, right, they're really sticky. But after a few times, they're full of all the stuff that was whatever it was that he was sticking them to. And so those molecules were kind of being brought into that stickiness. That's how an amoeba eats. It gets its stickiness and it brings that stuff in. But then you can also apply that to the way they move. Because when they find their food, they just throw their nastiness and then they bring themselves to it. Okay? So if I was going to narrate this whole situation here, predator, prey. So an amoeba and a bacteria. The lion and the gazelle. And the lion sees the gazelle and it comes and it consumes it. It's engulfing it through the process of endocytosis, more specifically because it's eating, phagocytosis. And now it's inside. It's eating it. Okay? There was no arms or legs. There was no ripping and shredding of anything. He's just going to get himself a bacteria. I just need you to understand phagocytosis and be able to apply it to an amoeba as well. This is the end of this chapter. This is the end of this unit for the second lecture exam.